In class, when we talk about mutations and their role in genetic disease, there's some very important bits of terminology that we need to use to have in common to separate all the various important issues to make sure that we keep them straight and build a really solid case for why our particular mutation is the one that's actually causing the disease. Pathogenic, that is, the definition of that word is disease-causing, is really kind of the gold standard that we're going towards. That is, you know that it contributes mechanistically to the disease. That is, you know, for example, like in the pathogenic mutations with the hemoglobin Woolwich and the like, where you know that there's a mutation in a particular gene that causes an alteration in the hemoglobin protein and how that leads specifically to disease. So pathogenic is what we're going for and we'd like to know, but we don't always have complete evidence for. One step down from pathogenic is implicated, where many diseases are, because it's enormously difficult to be certain of these things until a large amount of evidence has accrued. But implicated means you've got a bunch of good evidence that it's pathogenic, but you're not 100% sure yet, and so you want to make sure to make a distinction between, yeah, we really know for sure, like in the case of the beta-globin mutations, where we really know it's pathogenic, and implicated where we've got strong evidence but not conclusive yet. And then we have associated, which is even one step down. So we have some strong suspicions. That is, you find this mutation much more often in diseased people than in matching normal people, but you don't have any other information than just that. But that's a good place. Oftentimes that's where people start, and as you collect more information, you move towards implicated, and if you really collect a ton of information, you get towards pathogenic. So those are three things that form a spectrum. And then there are two more bits of terminology that are also important to keep straight. So damaging means it alters the level or biochemical function of a particular protein. So for example, you have evidence that the mutation actually alters the function of the protein, that you can study it in the lab, or you've studied the protein from diseased people, and you know that this mutation actually messes up the protein. It actually alters its function. It doesn't just change an amino acid, but actually changes its function. And the last one is deleterious. That is, you expect that it would reduce reproductive fitness. You don't necessarily know why, but somehow it injures the organism in some way, and so should therefore be weeded out by natural selection. So it has some injurious function on the organism. And so then damaging and deleterious are actually really two independent things. One says it messes up the protein, and deleterious says it actually hurts the individual. That is, you might imagine cases where it will be one but not both. So those are other important terminologies, whether you're actually damaging the function of the protein or it's being deleterious, that you're actually reducing the reproductive fitness, that is, you're injuring the health of the organism.